Thanks, Kim. Can you hear me? Yes. Okay. Uh, so I have to confess I'm actually not in Michigan right now. So I'm back in my hometown, which is Stephen, Minnesota. It's a little farming town. And can you see my cursor if I move up here? All right, more thumbs up. Yeah, so th this is where Stephen, Minnesota is. Now, if you've ever been to Minnesota, this is what people imagine. It's the land of 10,000 lakes and it's beautiful. It has trees and canoeing and everything. That's the part of Minnesota that's over here. So the part of Minnesota that I grew up in looks like this. So it's a flat expanse of prairie and farmland. This is from the Wikipedia page. So I grew up in what's known as the Red River Valley. It's one of the flattest expanses of land in the world. It kind of looks like Death Valley without the heat. In the region I grew up, the river, which flows north, which is kind of odd, it only drops an inch and a half per mile over the entire expanse. Now, why that's interesting for automated science, one, if the internet cuts out on me, you know why, because I'm in northern Minnesota. Uh, also, people immediately saw the opportunity that you can do a lot of farming here, and not just farming, but these things called bonanza farming, where there's huge machines, they process thousands and thousands of acres, all of the same type of crop. And industrialization and mechanization and automation came very quickly to this part. I remember 25 years ago when I was in high school, we already had self-driving tractors made by John Deere. It's a little easier to do that than self-driving cars because there's not a whole lot of things to hit and run into, and there's little traffic when you're in the middle of a field, but automation seems to happen pretty quickly up here. Okay, so today we're going to talk about putting AI together with automation. So you've had a great summer school so far talking about AI. Kim gave you a little teaser about the automation stuff that's happening at PNNL. I think PNNL and EMSL are really the world leaders in putting these two things together, both the hardware, the experiments that happen, and the AI that goes with it. The big reason that we want to put these together is at some point our AI models have to interact with data. And I don't think we have enough data yet to do all of the things that we've promised in terms of AI. So if you're training an AI model like the ones that you've talked about over the past couple of days, I view it as this formula where you need three things. You need to have a model that's powerful enough. You need a transformer model is very powerful. You need a diffusion model, something that can handle the type of nonlinearity that you think is in your system. You have to have the compute power to train the model. For some cases, a big GPU cluster. Other cases, like with our instance, you can train it all on a normal size CPU or a laptop. And then you need data. If you don't have any one of these three things, you're not going to actually have an AI model. Now, what's interesting is that for each one of these, they follow their own sort of return on investment curve. So normally you get a big return on your investment early on, and then eventually it saturates. And the question is, for each one of these three things, where are we on this curve? Now, speaking just about my area of microbiology, so not all of science, not even all of biology, just my subfield, I think we're pretty well set on compute power. So we normally aren't limited in the size of machines that we have for computation. We also have pretty good models. We have models right now that can learn English. We study microbial metabolism, so metabolism is challenging and intricate, but I don't think it's as intricate as human language. So I think our models are generally good enough. We need to do a lot of work to translate those into biology, but the models are there. What we're missing in microbiology is the data. I don't think we have the data yet that we need in order to train the types of models that we want. And here's some actual data to back up our lack of data. So our lab took every paper that's ever been published on a bacterium, and we downloaded them all and we analyzed what species that what species that paper studies. It turns out that half of all the papers that have ever been published in microbiology just study one of 10 species. So E. coli is number one. That's the best studied species by quite a bit. Staphylococcus aureus is number two, and then it drops off very quickly. So much so that 90% of papers study just the 1% of microbial species, and 3/4 of all the species that we've discovered and placed in the tree of life, we've never published a paper on them. So if you're going to train a language model, something to say it's going to read all the literature and then synthesize all this knowledge, there's really nothing for it to synthesize for most of the bacterial species out there. This problem, interestingly, is getting worse. So if you normalize the number of papers we've published by the number of species we've discovered, it's gone down by about 30% over the last decade. So we're amidst an ignorance explosion right now where we know less and less about more and more species. It's going the wrong direction. So we need to collect more data, and I think this is where AI is going to come into place. We need to start using machines and scale up the acquisition of good data. Now, the problem, and what I'm going to spend most about talking today, is AI doesn't seem to be our problem when we start talking about the robots. We have many more problems actually connecting the AI to machines and then getting the machines to operate. And I can't see Kim right now in the room, but I'm sure she's laughing when I bring this up and say that the physical world presents a huge amount of problems, and we spend a lot of our time on that. This is actually called Moravec's paradox, where things that are very easy for humans to do turn out to be hard for machines and vice versa. So if it's easy for a human, if we don't think about it very often, if we do it instantaneously, like interact with the physical world, tough for AI to do. Something that we really have to think about, like taking an exam, for instance, that actually turns out to be much easier for AI. So here's a couple of examples. A few months ago, I took a trip from Ann Arbor, Michigan down to Indianapolis for a conference, and I needed to plan out the route. So to do this, I go on to a map service. I think I use Google Maps in this case, and I said from Ann Arbor, where I work, to the hotel in Indianapolis, what's the best route? And within a second, it told me this is exactly the route that you should take. These are all the turns that you should do. That problem has been solved by AI for decades, figuring out exactly what route you want to do. Now, that's hard for me to do as a human. If I had to plan this out, I'd have to pull out an atlas, I'd have to sit down, I'd have to look at all the routes and decide which way I want to go, think about the traffic. It would take me several minutes to plan the same route. It's instantaneous and solved problem for AI. But after I get the route, uh, I get in my car and now I make thousands and thousands of instantaneous decisions that I'm not really thinking about. I've been driving for a long time. I don't give 100% of my brain power when I'm driving. You can think about other things while you're driving. Some people think about lots of other stuff when they're driving, as I noticed on the road. But self-driving cars, even though driving down the road is something that every human is capable of doing, that problem has not yet been solved, right? It's easy for humans. It's relatively easy for us. We don't have to apply a lot of mental power. It's largely instinctual once we learn, but it's still very difficult to do. And this is the paradox. Another one is that Amazon is very good at predicting what you're going to buy. So Amazon, in many ways, can pre-ship things to fulfillment centers. They can show you recommendations. So trying to come up with a gift for someone, I have to think a long time about it, but Amazon is very good at suggesting to other people what they might want to buy. However, Amazon in their workhorses has in their warehouses has tasks that they have not solved yet, physical tasks like picking things out of boxes and placing them on shelves. Right? This is a problem that's very easy for humans to grab something out of a box. We don't even think about doing that. But this is still a holy grail for robotics and automation research is how do you do that last step? The grab and grasp problem is still unsolved, even though from a cognitive standpoint, it's much easier for humans. So we have the same issue when we talk about applying science to robotics. I think now the models in some cases have shot ahead of what we can do physically in the lab, simply because the models focus on cognitive tasks, and in many cases they have better data. This is a news story about OpenAI. This, if you look at the date, this is from 2021. So OpenAI used to work on this grasping problem. They had a robot that could solve a Rubik's Cube using a robotic hand, and they actually shut down the robotics research program in 2021. And they said they are going to shift their focus instead where data are more readily available. They didn't say it at the time, but this was to focus on language modeling, and we know how that story ended. OpenAI did very well. So they're a very capable AI team, but they decided that we're not going to do robotics. We're going to get ahead faster on language modeling simply because we have the data. All right. Now, I think recently OpenAI has started to reinvest in its robotics program, but if given the two, they thought they could make better progress on something that was entirely virtual. So why is this so hard and where does automation come into play? I want to, for the majority of today, take you through a cycle where we've managed to figure out what processes are hard and what are difficult and how we can reason through what exactly we're trying to do in the lab and with robots. And it all goes down to figuring out what it takes to do science and which parts are easy to automate and which parts are hard. So let's talk about science to begin with. All of science begins with some sort of prior knowledge. You read the literature, you went to school, you interact with the physical world, and you have some preconception of what the world is like. That's the prior knowledge that you begin with. From that, you define a problem. So, based on the literature, I would say no one has really studied in a bacterium what this GAL-KT gene does. I believe that it affects growth on raffinose, right? So, I can define a particular problem. We'll tell you a little more what we mean by problem definition, and then we form a model. Now, usually biologists don't form mathematical models like you've talked about in the summer school. They form a mental model, a qualitative or sort of a logical model to say, here's how I think the system is working. And from this model, then you can come up with an experimental design. So a hypothesis is really just saying, if I work through the model in my head, here is what I expect the response to be. If I do this thing, if I set these inputs, here's the response that I'm going to get. For this example, if I give it raffinose and I delete this gene, it's not going to grow. That's my hypothesis, is formalizing a prediction of your mental model. So we come up with an experimental design, and then we say, well, now I'm going to carry this out. So I'm going to use some sort of protocols. Humans, again, usually plan this out in their head, or they find protocols that are pre-existing, and then they compile them into laboratory operations. They say, if I'm going to do a growth assay, I need to pipette this amount of liquid into this well of a multi-well plate. Humans usually do this on the fly. For robots and automation, we do this more formally. We write our protocols into a language, into instructions that the robots can understand. The robots run in the lab and produce some raw data. We process the data by comparing it to controls and subtracting the effects of blanks, and we get something that looks like actual information. And then at the very end of the cycle, we can take that new information and we can give back to human knowledge. We can add one more paper to the pile of papers and say, here's what we found from doing the experiment. So this is the cycle of science. It starts from knowledge. It goes through problems, mental models, designs, running in the lab, and generating new data, and then back to knowledge. So if we want to automate the scientific process, if we want to do this without humans, we have to remove humans from every step in this loop. We have to find a way to get this loop to turn without using human intervention. That's the challenge of automation. And you find that some of these steps, it turns out to be a lot easier to automate than others. And we can talk about that in terms of why things are difficult or not. Okay, so we're going to focus on this loop for the rest of the presentation. This system of formalizing the knowledge as this loop, this was developed in collaboration with two other individuals, Ben David, who's finishing up his PhD here at Michigan, and then Mark Hendricks, who is down the road from PNNL in the chemistry department at Whitman College. Okay, so when we talk about autonomy or doing this loop without humans, there's going to be different levels at which we can take humans out of the loop. So this is an autonomy chart from the Society of Automotive Engineers where they looked at self-driving cars and defined that there are actually 6 different levels of autonomy that you can have. Everything from level 0, which is no autonomy, up to level 5, which is a completely self-driving car that's 100% autonomous without humans. All right, so they've defined these nice levels in between, and we thought we should do the same for science. Like, what are the different levels of autonomy that you can give in a scientific experiment? So let's do that, and we're going to start with level 0. So level 0 is actually over here, which is also known as laboratory automation. So adding robots to this, just buying a liquid handler or buying some of the robots that Kim talked about, really automates this step. They're programmed by humans, so they take a set of instructions from humans and they produce raw data, right? So humans are replacing humans with robots in the lab is just level zero, and all it automates is this step here. It doesn't actually allow us to go through the cycle any faster by itself because this isn't a closed loop of autonomy here. It's one step. So if you have scripts written for your robots, if you have instructions ready to execute on the robots, and you have humans that are ready to process the data that comes off them, then automation is your bottleneck, and automating this step is going to make this loop go faster. If not, making this step go faster is just going to deplete the number of protocols that you have ready to run and pile up the data over here, and you're still going to be waiting for humans through the rest of this loop. So level zero autonomy is fantastic, but it doesn't necessarily speed things up. Buying a robot is not going to make your lab go faster in the long run unless you have a pile of experiments ready to go and a pipeline set up in order to process the data. Okay. Now, people have actually studied what happens when you bring robots into the lab. This is a fantastic paper. It's a study of a number of synthetic biology biofoundries that were set up in the United Kingdom. And they studied the researchers and their interaction to level zero autonomy, as we call it, or just adding robots into the lab. And one of the things they notice is that it completely changes what you do. So when you think, I'm going to get a robot in the lab, let's say get a liquid handler, before I had to spend all this time pipetting. And after we get the robot, I'm not going to have to pipette anymore. And that's right. You'll instead spend your time staring at the liquid handler and making sure that the robot is doing the pipetting for you and doing it correctly. So you go from doing the robots to babysitting the robots in essence after level zero comes in. And a lot of the people they interviewed talked about how they did not realize that when you bring in autonomy, bring in level zero automation, so much of your time is going to be spent focusing on things that are automatic to humans. Did the liquid actually transfer? Did it move the plate and put the plate in the right position? These are things that humans do instantly and we learn how to do very quickly that you're now going to spend weeks and months of your time trying to get a robot to do for you. So it's very difficult and it changes what you do. There's also problems that it creates because the robots only automate certain parts of the process. So this is a picture of the deck from one of the robots that we have in the lab. And when people usually think about automating and programming the robots, they're talking about how do we transfer liquids from some of these slots where we put in conical tubes to these positions in the well. They think about what happens on deck. And that's an important part of scheduling these robots. But really, we have to get all this stuff onto the robots before it starts. So how do we get chemicals off the shelf into the liquids that can go on the robot? How do we get stuff out of our freezer, out of the fridge? These are pictures from the actual lab. This is still all done by humans. And having a very flexible lab, buying a robot and not automating the material handling that goes with it as well, now all of this becomes the bottleneck. We still haven't solved the problem of dealing with all these tubes and all the boxes in the freezer just because we have one robot. Okay. The big thing that I alluded to before is that simply buying a robot is not going to speed things up in a lot of labs outside of very repetitive labs like clinical settings because experimental design is the true bottleneck. So the same paper I showed before asked scientists what do they spend most of their time doing? And just as much as collecting and analyzing data, which is downstream of the robots, and designing experiments, which is upstream of the robots, those are the two biggest time sinks. Doing laboratory work, doing the work that is going to be replaced by the robot, that came in as number three, right? So the big issue that we have is coming up with new experiments here, this part of the loop, or processing the data over here. And robots by themselves aren't going to do it without some other models that can do the experimental design or the data processing. So that was a bit of a pessimistic view of just the robots themselves. They are fantastic. They are a lot of work, but to really speed up the entire cycle of science, we're going to have to do something else. We're going to have to get all the way around this loop. Now, the first way that we're going to get all the way around this loop is to actually cheat. So at level one autonomy, what we can do is if you have an experimental design, we can do all the things that Kim talked about when she said we're going to do the orchestration, which is scheduling the robots, writing protocols for the robots, taking what the scientist wants to do, which is the experimental design, and translate it into something that can be run at this level 0 automation by a robot, and then translating the data handling, which is something that you talked about in the previous days in the summer school. Now, if we can cut across the circle right here, if we can go from processed data back to the experimental design, right, then we can have this thing run without a human. Now, the problem is, what is this cutting across to? So the first level that we can modify, which we call level 1, is all we're going to automate is just verifying if the data are correct. In essence, we're going to automate the quality control plan. So if we have the process data, we can say, do these pass our quality control standards? If they do, then we can say, all right, humans, we're ready for you to look at the data. If they don't, we can re-queue up the exact same experimental design and run it through again. So this would be a huge time saving that the robots would run. They would try and produce data. If the data are not good, the robots will immediately run the experiment again another time and only alert the human when you actually have good quality data that comes out. So we call this level 1 autonomy. If you can get all the way around this loop, automating your orchestration, the initial data processing, and the QC, then for the first time, you can do useful work without a human. It's just that you will keep running the same experiment until the data are good enough. Okay, so we actually developed a level 1 system in the lab to run growth experiments for bacterial growth assays. So here's a bit how it works. We start with this experimental design. We have a set of runs that we want to execute. We have, for instance, 3 different media ingredients. We have different concentrations. All this gets put into the scheduling system. So we have a big computer that takes in those experimental designs and converts that into instructions for robots. So it first goes to a set of robots that set up source plates. So they take the tubes that we pull off the shelf. That's done by a human still. We put them on these robots and they move everything into 96 deep well plate format. Those plates get transferred to a set of other robots that actually set up the individual experiments. So we do very high combinatorial screens. So we have specialized robots that don't require pipette tips in order to do it. They're non-contact dispensers that actually set up the individual experiments in 384 or 96 well plates. The plates then go into other robots that add bulk media or that inoculate with the bacteria. We use separate instruments to inoculate usually because those instruments are a little easier to clean and sterilize once the bacteria have been through them. Then they go into robotic incubators. And the robotic incubators and arms move them in and out of plate readers where we monitor growth or fluorescence, or we can take microscopy images of it. And at the end of the day, we get data back corrected to the controls. We get the process data. Now, at this point, we can decide. We can do some quality control analyses and say, do these data look correct initially, or do we need to run this loop again? And if the data look bad, we instantly reject them, and then we run them back through the scheduler and repeat the same experiment. So humans only get bothered. They only get a message to say, you need to do something with this data when we know the data are correct. It's been through maybe 2 or 3 times. We have, if any data were missing, if a plate was dropped, it runs through again. If the standard errors are too high, we run again for more replicates. All of that can happen in a closed loop without requiring a human to make a decision about it because we have this level 1 autonomy in place. So in our lab, we started about 5 years ago. We could do 300 experiments a day through the loop. Right now, we can run about 6,000 experiments a day. And starting this summer, we're building a new incubator, which is our bottleneck. We're going to be able to do about 60,000 experiments a day. And again, the experiments run and run and run until they pass quality control, at which point they can exit the loop and we have to get a human involved again. Right? Automating quality control is very difficult. This turns out to be something humans are good at, looking at data and saying, I think something was wrong with this experiment, but there's a lot of subjectivity, a lot of domain expertise that goes into there. Here's one example with one of our robots that we have. This is a Formulatrix Tempest. I think one of the robots that's going on the system at PNNL. It has these microfluidic chips that do the dispensing here. Each chip has 8 different nozzles, and we actually QC these chips. We have 6 chips on a robot with 8 nozzles. Twice a day, they go through a quality control step where they dispense dyes of a fixed volume, and we put them into plate readers, and we can detect when they're dispensing a little off. So here we see this one chip, this one nozzle. On one day, it seemed to have a clogged nozzle, so we have to go in and clean it or replace the chip. So we have a whole QC system just to see, is the robot operating correctly, because we can't have a human watch every pipetting step. Humans, while they're pipetting, will usually know when something didn't happen or the tip was loose or something. We have to have an entire system, a statistical process control in place in order to flag things and say, no, you need to run through the cycle again. This QC has failed in this batch of experiments. Okay, so that's level one. We can run through a cycle and repeat a single experimental design until we get the correct data. And this requires automating the scheduling, the compiling, the initial data processing, and most importantly, the QC. The QC is the thing that allows us to jump across the cycle and actually turns it into a loop so that it can run. So for level one, the QC, I think, is the most important step. You can move one level up from there and say, well, I really would like to not perform the same experiment every time. That's useful. That's sort of the first level that needs to happen, but we should be able to propose new experiments. So at level 2 autonomy, as we call it, you take the process data, and thinking about those data, an AI agent is able to say, here is a new experimental design. Now, it turns out that what actually happens is we go through the model. We take the data, we use it to retrain our model. We translate from a mental model now to a mathematical model, and then use the model to plan what are the informative experiments going next. So now we have a cycle that runs this way. We can change the experimental design and run it through again. Right? So these work in sort of a nested fashion. We originally come up with an experimental design. It runs through the cycle. Oh, maybe it failed QC, so we need to run again. Now it passes QC, so we can run through this outer loop, come up with new experiments, and go through the inner loop until it passes QC, and then one pass through the outer loop. We don't really skip the inner loop. The inner loop runs every time, but every time that inner loop passes QC, we're allowed one pass through the outer loop again. So this inner loop becomes more and more critical as we add these nested loops to it. So let's talk about this level 2 design. Level 2, the input to level 2 is this problem definition. So here's what you need to do before you can do level 2 automation. These are the things the AI agent can't do. They have to be done by human. First thing is the human has to say, here is the actual problem you're going to solve. Here's the factors, here's the ranges of each factor. So what are the concentrations you're using? If you're changing oxygen concentration, is it discrete levels? Can you do just anaerobic and normoxic? And then what levels you're going to do in between. And also, what are you measuring? Are you measuring growth? Are you measuring expression of a protein? Then you have to have a model that says, given these inputs, can the model predict what the output of the experiment is going to be? Given these conditions in the media, the model predicts here's what the growth rate is if growth rate is what you're measuring with your system. You need an objective to say, am I trying to maximize this? I'm trying to find the maximum growth rate, or trying to find the minimum growth rate if it's a pathogen I'm trying to kill. Or are you trying to characterize? Say, I just want to learn how all of these different factors affect the expression of this gene. I'm not trying to find the highest expression or the lowest expression. I just want to learn the mapping between inputs and gene expression. And the last thing is you need a plan for selecting the experiments. And in the AI literature, this is called a policy, to say, given the data that I've currently collected, what is the next most informative experiment that I should run? So after the human has defined all 4 of these things, then you can run at level 2 automation. I'm going to show you an example right now of how we do this inside the lab. So here's our example. We're going to study the effect of amino acids on growth. So we're going to take the 20 proteinogenic amino acids. They're going to be varied between 2 concentrations. Either they're out of the media, so none of the amino acid, or at 10 millimolar. We're going to measure growth, which we're going to define as sort of the final optical density at 36 hours. So you can use more complicated phenotypes to measure, but this is one we're going to use in this example. For our model, we're going to use a neural network to say, given a set of the 20 amino acids in or out for each amino acid, it's going to try and predict the growth of a particular bacterium. And then our objective is going to be to characterize. We're not looking for maximum or minimum growth. We want to learn this entire relationship. I want to know what amino acids are going to lead to what growth inside the media. And for the policy, I'll explain this a little more later on. We're going to do two things. We're going to do random experiments, and then we're going to try and dig down and find small media that have growth. We also don't let the model repeat experiments, and I'll tell you why in a little bit. Okay, so this is our problem definition. This is come up by humans. Said this is going to be a interesting biological problem based on our knowledge of the bacteria, and we set these parameters up, and then we turn it over to a level 2 system. So to start off with, the humans don't really have any idea what amino acids are needed for growth. And even if they did, we might not want to bias our AI system by saying, here's what the human thinks the system looks like. So we start all of our experiments entirely from a blank slate. We start with a randomly initialized model. We don't have any prior knowledge put into it. We don't try and take anything from the literature if it exists for the bacterium. We say, why don't you just pick random experiments and do that in the first pass? So on day one, we pick 336 random experiments. That's a nice number for the robots after we add all the controls and blanks to the plate. And it picks the experiments by varying which amino acids it's going to put into every media. So I'm showing you these plots here. It can take out between 0 amino acids and 20 amino acids. So this would have everything in it, and this would have everything taken out of the media, right? It chose to do 6 experiments here that have 1 amino acid missing, right? Each dot is a separate experiment. If the dot is filled, the model predicts that the bacterium is going to grow. If it's an open circle, it predicts that it is not going to grow. And after we get the data back, after these experiments are run by all the robots, we can color them and say if they're a black circle, then the agent was correct. And if they're a red circle, the agent was incorrect. So this is day one. It's picking these experiments entirely by random, and there's a lot of red circles. In fact, about half of them. It's almost a coin flip to say, did it grow or did it not? Because the model really has no idea if it should grow or not. So a lot of things were incorrect. Now, how we pick these experiments after the first round uh, is to say, well, we know that it grows when you give it all 20 amino acids. That was prior knowledge that we had. And if you take them all away, it's not going to grow as well. And somewhere in between, in this 20-dimensional space, there is a line where it goes from growing to not growing. And we believe that that's a very complex shape there. There's a lot of combinatorial logic in amino acids that determine when it switches from growth to not grow. And we want to map out that growth front. So one thing we do is we just have a random policy. So we start with 20 amino acids and we start taking amino acids out virtually one by one until the model predicts that we can no longer grow. That means we've hit this growth front. It grew right here and we took out a single amino acid and now it doesn't grow. We say, well, those are interesting experiments to do right at this transition, right at the boundary from where it grows and doesn't grow. So we should add those to the list of experiments to run. But we got there by simply a random walk. Another thing we do is we use a technique called rollout, where we're trying to dig down and say, give me the smallest media that is predicted to grow, but if you take out a single amino acid, it's not going to grow. And when we find those, then we also add them to the experiment. So we plan about half of our experiments randomly and half using this rollout, this exploitation type policy to find interesting experiments. Now, the agent scores points for getting the experiments correct. That's what it wants to do. It wants to train its model, maximize the information that it gains. So we can't let it repeat experiments because otherwise it would plan 336 experiments. It would get the answer by running the physical experiment, and then it would just replan the same 336 because it knows the answer. Now it's going to get them all correct. So we don't allow it to repeat any experiment. It has to come up with new experiments every round to avoid the agent from cheating and just repeating the same thing every time. Okay, so this was the day one again. This was planned entirely by just picking random experiments with an untrained model. These data come back, we update our neural network, and now we go through those two policies to pick a new round of experiments. We do this every single day. By day 3, you can see already the number of red circles has gone down quite a bit. So the agent is learning here. It's much better at making predictions about what's going to happen in the next round of experiments by collecting data. You see, it learns a lot about the media that have lots of amino acids, and now a bunch of the red circles are actually pushed down. When you start taking away a lot of the amino acids, that's where it's still having trouble, and that's where it's pushing more of its experiments. So this is day 3. Here showing day 5. It actually was a regression in the accuracy. This happens even to normal scientists partway through grad school. You thought you understood the system, you get some data, and now, oh, wait, that's surprising. And I don't understand the system as well. But by day 9, it's usually recovered. And now we're past day 10, and you can see we're over 90% accurate. So the model is telling you 90% of the time before you run the experiment what the answer is going to be. Will it grow or will it not grow? And the model sort of stabilizes there. So we stop the agent, and now the agent knows, given any combination of amino acids, even ones we haven't tested, it can tell you with over 90% accuracy, is it going to grow or is it going to not? Okay. Now, one of the things that we learned from doing this is we originally thought we were going to be super clever in how we picked experiments. So we have 2 to the 20 or a million possible combinations of amino acids. How do we get through all of those combinations or enough of them to be able to train an accurate model? And we thought, well, there's some tricks we can apply here. So let's pretend that we have this subset of amino acids here. I'm showing 6 of them. If you take out one amino acid, that defines 6 possible media that you can make. So these are single amino acid removals. I've represented them as sort of a tree here. And then for each one of these experiments, I can take out other amino acids, and now I have all the pairs. So it branches again beneath it, and this keeps on branching until there's a million leaves in the end of the tree, right? Because there are 2 to the 20 possible unique combinations that come inside this tree. Well, what we thought to be clever is that if I find a media doesn't grow, if I'm walking down this tree and all of a sudden the bacterium stops growing, if I take out more amino acids, it's not going to bring it back to life. If it can't grow in this media, taking more things out isn't going to bring it back. That was our idea. We call this metabolic convexity because it's related to mathematical convexity, which means that every time we hit a media that doesn't grow, we can just stop the tree. We don't have to explore anything below it because it's dead. It's going to be dead. It's not going to come back to life. We thought this was really clever. This is how you solve mixed integer linear programs where you actually have convexity. You can bound how good the solution is going to be and solve these really difficult challenges. This is great. It turns out that this is wrong entirely. On the first day, it found a condition where here's a media that doesn't grow, you take something away, and it comes back to life again. Right? Bacteria don't have metabolic convexity. That's an oversimplification. And by using this exploration strategy, we were going to miss a lot of very interesting things. So we try to get way too clever with the search space and say this is a fancy policy. It'll be very efficient from a theoretical standpoint, from a modeling standpoint, and then biology sort of made fools of us all. So simple policies, simple models is the way to go when you're driving experiments in ARC. As a first pass, should be focused on simplicity. Okay. So that's our level 2 autonomy here. We've come through this loop now. We keep the model the same, so we haven't changed the problem definition. We have a model that gets updated. We create a new experimental design. And every time we do a batch of experiments with this Bacter AI system that we have, it runs, we finish a set of experiments, it goes through QC. If the QC fails, we immediately kick it back and we run the experiments again. So sometimes at the end of a day, based on QC, we just rerun the same experiments from the day before because the experiments look off, something went wrong. We run QC with the same experimental design until we get a batch that passes QC. Then we go out to the model, update the model, we replan the experiments by using this policy, and then we send those experiments out. They go through this first level 1 cycle until they pass QC and back out to the model, and it iterates until we stop and say, all right, now it appears that the model isn't learning much anymore. The model is already 90% accurate. We can stop running through this cycle. Okay. Now, what this doesn't do for us is create any knowledge. So knowledge is up here. This is what humans can interpret. We are short-circuiting out knowledge by just training a model. there's information that's been gained, but it's all locked inside this neural network. So separately, we need to take our data and actually interpret it. We need to extract knowledge for humans because this wasn't included in our loop. To get the system to run, we had to bypass knowledge. So now we have to go back. So we actually built a second part for our bacteria eye system. This is the first loop that we talked about where we're collecting data in this level one system. And now we have a different AI agent whose job is to extract information from the data and from our models and create knowledge for humans. And we do this by constructing sort of logical rules that tell us what the amino acid requirements are for the bacterium. So here is sort of the output from one of our second agents, this knowledge generating agent. So after Bacteria I did about 2,700 experiments, that's a tiny fraction of the million possible combinations of amino acids. It says this is the rule that you have. You need isoleucine, leucine, or valine, and you need glutamate or glutamine, and you need leucine or proline. And we were pleasantly surprised by this. If you're have a bit of a biochemistry background, you know that this kind of makes sense. These are the ILV amino acids. They share a lot of pathways. Glutamate and glutamine are separated by one enzyme. So having one or the other, that oxytrophy makes sense. This last one was a little confusing to us, especially because when we asked a graduate student who had built a metabolic model by hand of this bacterium, he looked at a set of experiments he did using the robots, but not planned by the robots. So he did 422 experiments. He came up with this oxytrophy. He said, I think it's the ILV amino acids or glutamate or glutamine. And he did not mention anything about leucine or proline. So the question is, who is right? Was the graduate student right, or was the Bacteria AI system right? And it turns out that the graduate student was right. If you look at the 422 pieces of data that he had, there's no leucine or proline oxytrophy that you can gain from that information. However, if you look at all of the experiments that Bacteria AI did, so 5 times more experiments than the graduate student did, then this oxytrophy shows up. So it only shows up after you've taken out a lot of other amino acids. They have this second oxytrophy. So even asking who's right, what is the gold standard by which to assess our model depends on the data set. It wasn't that the graduate student missed this oxytrophy, it's that the oxytrophy does not appear in his limited data set because he needed a data set small enough that he could analyze it by himself. When you ask the AI agent to collect a larger data set, an unbiased data set, then you see these oxytrophies that the humans missed. So I think this raises a lot of interesting questions about what is our gold standard? What are we training and benchmarking our models on? Are they human generated data? And what are those models missing because of our biases, where we can only focus on small data sets and analyze them? Okay, so we have this level one, the QC cycle. Level two is this automated science cycle, sort of a Bayesian optimization cycle is a technique that's used a lot to run these level two cycles. The last thing we could do is sort of go from process data and actually change the problem that we're interested in studying. So rather than go straight to the model, we can say, I have some data now. Should I be tweaking the problem, not just the experiments I'm using to solve a particular problem? Hey, so for an example of this, we had Bacteria say, well, we've learned some things about this bacterium. Maybe we need to switch and learn about a different bacterium instead. So Bacteria started doing this at the end. It said, well, given what I've learned about Streptococcus sanguinis, so that's the bacterium shown here, right? Can I use that information to help learn about a different bacterium? This is called transfer learning in the machine learning community. And it actually works out really well. So if you start with sort of a blank slate model where we don't know anything about the bacterium, it took about 7 days and 2,300 experiments before we got a good accuracy model for Streptococcus sanguinis, one of the bacteria that lives in your mouth. But if you take this model for sanguinis and use that as the first model to learn about Gordonia, even though we know it's wrong, we know these bacteria are different, we're just using the knowledge from the first one and then learning to fix the model. Now, not only can you learn about Strep Gordonii, but it takes a lot fewer experiments, only 4 days and 1,300 experiments. And we got a really good model in the end. So with transfer learning now, we can do this level 3 cycle where we're not just changing and picking experiments, we're actually picking a new problem that we can study with the bacterium. Here's another example of this transfer learning process. If you take that same strep sanguinis model and study amino acids, there's actually about another 19 ingredients in the media that we didn't touch in this first study. There's vitamins and minerals and cofactors and carbohydrates. And what if we want to learn about all the metabolic dependencies of those as well? So we can do the same thing. We can transfer in the knowledge from amino acids into a larger problem, into one now that has 19 more variables, so 39 variables rather than 20 variables. And that takes only another 7 days in order to uh, train a model to about the same level of accuracy. This second problem is 500,000 times larger. It went from 20 variables to 19 variables in a combinatorial space, which is a huge explosion, but it takes only a linear increase in the number of experiments that you need to do because we're transferring in this knowledge. And we see this all the time with these automated systems that the learning rate seems to scale about linearly to uh, the number of variables that you have, even though the space that you're exploring scales combinatorially. The last sort of level that you could think of, we have level 0, 1, 2, and 3 is to really do the same thing where you're taking data and coming up with new problems, but if you use human knowledge as an intermediate. So if now instead of reasoning about things using neural networks and models and things that humans can't crack open and interpret directly, if you had the data get processed into human text, or if you had an agent that wrote a paper and then other agents read that paper and turned that back into a problem, then I think you're doing something a little different where you're using human knowledge as the intermediate. Here, we just use computational knowledge, knowledge locked away in models as the intermediate. Right. So I don't have an example level 4. This is not something that we do a lot of in our lab yet. But I think this is the direction where agentic AI is really heading, is being able to keep humans in the loop as you're designing this last step, as you're taking your data and turning it into new problems. I think that's very exciting. There's a lot of people at PNNL, some of whom you heard about with the language modeling, that are working on this problem. I think it's going to be very exciting for science. So the last thing I want to mention here is that you don't need robots in order to do autonomous science, right? So here is normally how we do science. Humans run experiments that humans have designed. So we take data and say, this is our next experiment, and then humans go in the lab and do this. This works really well. It's generally pretty slow and not super efficient. The designs humans come up with aren't that great. Here's what I talked about today instead, where we have an AI planning experiments and then robots running them. And this is what a lot of people think about for autonomous science and automated science. And this works very well, but there's a couple other things that happen in between. One, you could have humans plan experiments and then robots run them. This is what a lot of people with laboratory automation do. The robots run the experiments, but they rely on the humans to interpret the data and program them. This, I think, is the sweet spot over here. So if you can use an AI to plan experiments and then have humans still run them. And I think this is a very underutilized permutation of this automation science, is use the power of the AI to analyze the experiments and predict the next ones, but then have humans still go in and do this. And we still do this in our lab. We have, all sorts of robots, but some experiments we have not automated. They're too expensive, they're too difficult. We don't have that type of robot. So we still do studies where we use models to help pick the experiments, but then humans go in and perform all of those experiments by hand and return the data back to the model. And you shouldn't be afraid of this. I think this is a much easier start for people. You don't have to manage all the physical challenges in the lab. It's much more accessible. There's more people that could use an AI model to help plan their experiments than could afford to buy robots and know how to train them. So the future, I think, is going to be very decentralized, where we have lots of people doing this model guided science. And I am always encouraging people to try the models, even if you don't have the automation. So, sort of as a wrap up, here's the 4 things we've learned from doing these different levels of automation. One, if you're doing level 0, if you're working with the lab automation, as that study pointed out, be prepared that you're going to spend a lot of time focusing on pipetting. Your PhD could be focusing on getting robots to do something that a human could do very efficiently. It's mundane work. It's a lot of engineering work. It's so cool when it works, but it's a tough road. On this level one, the big step is automating the QC. It only runs in a closed loop. You only get the savings and can have this run overnight if you focus on automating QC. And unfortunately, that's one of the last things that people think about for automating. They want to focus on the scheduling or the data processing, not that question of, do we reject this plate and queue it up again? If you solve that problem, you can run in a closed loop by keeping the design the same. For level two, I mentioned this before, when you're starting out, keep the policy simple, keep the model simple. All right. And worry about the complexity later because there's a lot of things you can do by just getting the experiments going and being surprised by the data. Once you get to level three, where you're designing new problems and you're coming up with new experimental designs, keep all that separate. The AI that designs problems, picks new problems, should be different from the one that picks experiments, should be different from the one that writes lab protocols. Those are all very different problems. And every time we try to use the same piece of software or the same system to solve all of those problems together, we end up re-extracting it and separating it again. Right. So it does not work to try and cross those levels of extractions. I think you should try and keep them separate. So with that, I want to thank you for your attention this morning. I want to thank all the members of the lab who did this work. Ben David, as I mentioned before, is a PhD student who did most of the work that I was talking about today. Of course, a thank you to our funding sources and all of our friends at PNNL, and happy to take any questions either now or on the Discord site later. So thanks.
Thank you, Paul, for that wonderful presentation. This is Abby Jerger. I'm taking over for Kim. Do we have any questions here in the audience? Otherwise, we can move to the online questions that we already have. Okay, we have one in the audience.
Thank you for the wonderful presentation. So my kind of question is just more of like generic microbiology question. So why do you actually consider amino acids as one of the critical factors affecting the growth of the bacteria? Why not carbohydrate or something else? Because I feel like combining all these factors could actually, like just like the metabolic intricacies when it comes to bacterial growth is very critical. So why do you specifically choose the amino acids as, the model to train on that could actually affect the bacterial growth in general? So
Yeah, so you're right that these bacteria, especially, we study bacteria in the mouth, and they have all sorts of oxytrophies. So that they have a huge number of dependencies, including carbohydrates. They're oxytrophic for a lot of vitamins, cofactors as well. We started with amino acids because it's the one thing that we couldn't predict very well with a mathematical model. So the graduate student that I mentioned, Keenan Jajaki, built a metabolic model for these bacteria. The model was very good at predicting which carbon sources it could grow on, which vitamins it needed, and especially in species to species differences in those. All of the models were uniformly bad at predicting amino acid oxytrophies. So the bacteria have full biosynthetic pathways for all 20 amino acids. So the model predicts that they shouldn't have any oxytrophies at all. It's entirely a regulatory thing. And we noticed trying to tease this out before we used the AI system that there were a lot of species specific differences in the amino acid oxytrophies, which was strange because all of these bacteria are close relatives. They're all streptococci and they all live in the mouth. So because we had that question, we said, we don't understand what's going on with amino acids. That's why we started there. And then you saw that after we finished with amino acids, then we expanded and said, well, now let's add carbohydrates, let's add cofactors. And there's all sorts of interactions between them. The carbohydrates affect the amino acids. The environment affects the amino acids. If there's oxygen, changes the amino acid oxytrophies as well, just as you predicted.
okay, thank you.
My question is also related to oxytrophy for this particular bacteria. As you mentioned, that after the vector I, the automation mechanism, uh, it predicted after 2600 iteration of bacterial growth that proline and isoleucine is important for their growth. Do you think it's because of the mutation happening in the growth of bacteria? That's why it's losing that ability, or it's just like genetically it's prone to happen that like bacteria would not grow after these many of iterations of growth?
Yeah, so one thing is that the experiments are performed in big batches in parallel. So and the bacteria are restarted every time. So it's not an experimental evolution where the same bacteria are passaged in different media over time. Because then, like you said, there would be mutations and things would change. We'd actually be learning a different target. So every day we restart the bacteria from a freezer stock and then grow again. So we're trying to learn about that initial wild type of bacteria. Um, so we've done 2,600 experiments on the same strain, started at the same point. We just did them in 11 or 13 rounds. Why they have oxytrophies, we're not really sure. One of the things is that, they live in our mouth. They're obligate commensals or obligate pathogens. So we have to feed them all the time. They can't live outside of the human host. So they've probably, because they're bathed in amino acids from our diet all the time, they've learned to just not use certain amino acids. Why spend the energy when you know they're going to be in the environment? That leads to some weird oxytrophies, like they require both methionine and cysteine, and we think that's a regulatory thing. They can't sense the difference for one. They just assume if one is gone, both are gone. Probably that means that the host is not eating. Um, so that's our guess as to why the oxytrophies exist, even though they don't have to be. We don't know why they're
Okay,
different.
I
Why
have a
this
question.
species.
Oh,
Oh,
sorry.
sorry. I was gonna say why this species and this other species that live together, why they have different ones, that's completely mind-boggling to us. So sorry, go ahead.
So I had a question more about the um like the process of automation and especially like the level 1, because I do a lot of this growing experience as well. And I feel like usually when you get like weird data or you're like, okay, I need to repeat this experiment, then you also need to do like some type of problem solving, like trying to figure out, okay, why are the results like weird or not what you're expecting. So can the robot like do this kind of thing when it needs to do like the quality control, right? So it can tell like, okay, these values are off, but can it do like this kind of like problem solving thinking like, okay, what was it that went wrong here?
Right. No. So in order to get an assay that's automated, we have to have it, reproducible enough that we think, all right, if everything goes well on the robots, this should work fine. Most of the time why things fail QC is because the bacteria had a bad day. Like that, for whatever reason, the overnight culture didn't come up right, and you see just sort of across the board, things are not growing well. So our approach to QC when we did these experiments was if something looks off, try it again the next day. And what we actually do is every day we take 10% of the experiments that we ran and we randomly we pick 10% randomly and we rerun it the next day and we plot them against each other and say, all right, if today's experiments don't match exactly last experiments, then something was a little off, and we run through that level 1 cycle again. So there's no troubleshooting that happens. It's just saying we can tell something is off, and you need to try it again and hope that problem goes away. And if it doesn't, if we go through that level 1 cycle 2 times and something's still off, then a human needs to go in and say, wait a second, what's going on? Now, what we've gotten better at is that our system actually now tracks everything that happens in the lab. Every vial that things came out of, every source stock, all of that's now documented. And the reason we do that is we hope eventually we're going to have a machine learning system that can try and do this troubleshooting just by association to say, wait a second, these are off. What changed? Did something happen? And maybe you'll say, oh, this stock has been in the freezer for 7 days, as opposed to 3 days when the normal experiments are run. That might be something that's interesting. Until we get all of that metadata tagged, which is starting up now this summer, we haven't been able to try and do that problem solving thing. It's all, oh, something's wrong. We're good at detecting something wrong. We're not good yet at detecting what's wrong. So that's a great point.